I always show this slide when I introduce my lab. So my lab is purely computational. It's a computational lab. We don't do any wet lab experiments. So that makes my lab project highly diverse and highly collaborative. And my lab mainly focused on using bioinformatics and a genomics approach to address some basic questions and also to facilitate crop breeding, like facilitated gene cloning, KTL mapping, and the marker development. And we also develop biological database and the computational tools that can facilitate large data dissemination, storage, and the mining. As I mentioned, the project in my lab is highly diverse. You can see the organism we are working on: tomato, cucumbers, sweet potatoes, and all kinds of fruit and vegetables. But I mean, my lab is still mainly focused on fruits and vegetables, but we also do other stuff like this penium is a freshwater algae, and also on viruses and viral vectors such as whitefly. I may miss some. I just realized. I'm sorry, Maria. We worked together on the fungal, a particular fungal. I did not use it here. And today I will mainly talk about the genomics work my lab has been doing for the last like 10 or 15 years. So the first one is reference genome. I mean, I think you all know that reference genome provides a valuable resource for research, basic research, and the breeding. It also can help us to understand the genome structure and the evolution. So we have been involved in generating the reference genomes of these many crops, fruits, and vegetables. We're also involved in population genomic research, like you do a large-scale genome resequencing, you call variants. So those variants can help you to understand the population structure, the differentiation between different populations, and the demographic history. It also can help you to understand or provide insights into genetic basis of crop origin, speciation, domestication, local adaptation, and the response to climate change. And we also, the population structure data can also help you to understand the genetic architecture of important agronomic traits. So in my lab, mainly focused on fruit quality traits and disease resistance. So that's kind of like you use genome-wide association analysis to identify some regions, genome regions. So we have been done or are currently doing for this number of crops. So as shown in this slide, you can see there's a huge phenotypic variation or huge phenotypic diversity within a species or within a genus. So the single reference genomes will miss a lot of information. That some genes, they are only present in one species or in one accession, but not captured by the reference genome. So that's why now we are focusing on using this pangenome approach to do this kind of analysis. This will bring your research or some discovery into another level that can be discovered using the single reference genome. So by definition, pangenome captures the complete genetic element within a species or genus because it uses multiple representative accessions to construct this pangenome. And this pangenome now is mainly using this graph-based pangenome approach. So this multiple genome, the sequence are captured in pangenome graphs. So each node represents a sequence. The age represents the relationship between these sequences. So the genome variations that will be captured in this pangenome graph. Not like the linear sequence, you only capture this one path. So now we have multiple paths. So when you do sequence alignments against this pangenome graph, it will search all passes to find the best alignment. It will improve the read alignment accuracy. It also helps you to identify structure variations. So using short reads to identify structure variants, that will give you a lot of false positives, especially false negatives, especially large insertions that were not captured by short reads. So we have been doing this pan-genome approach research on many crops. And one thing I would like to mention, we're currently doing the reference genome or pan-genome for sweet potato. This is a very special crop. It's a hexaploid. It's not diploid, it's hexaploid. And we are trying to build the phased, 6 haplotypes. Not like in tomato, in other crop, you just build one haplotype because they're highly homozygous inbred line. So, it will give you a lot of information when you're using this hexaploid phased assembly. And we are also using this hexaploid genome and deep resequencing of hexaploid sweet potato to check the allele dosage effects on the agronomic trees, and also the changes or the trends of allele dosage during the domestication process. Okay, that's it for today.